Okay, so I'd like to start by thanking Roberta for inviting me here tonight. It's always a lot of fun to come and talk to the public about the work that I do. So I'm a medical researcher here at the University of Toronto. And the work that I do in my lab actually is focused on a group of viruses that infect and kill bacteria. And these are known as bacteriophages or phages. So like us, bacteria can be infected by viruses and be made sick and even die from their infection. And also like us, bacteria have actually evolved a type of immune system to kill these viruses and to prevent themselves from being killed by them. And this is known as the CRISPR-Cas system. And of course, scientists have figured out how to remove the CRISPR-Cas system, this bacterial immune system, out of the bacteria and put it in a test tube and engineer it into human cells and animal and plant cells and use it to make very precise DNA edits. So CRISPR-Cas technology has really revolutionized modern molecular biology and biomedical research, and it's been uptaken by the scientific community at an absolutely astounding pace. So the first mention of CRISPR in the literature actually was in 2002. So what you see here on the slide is the number of scientific publications versus year. So first mention of CRISPR was in 2002, and all that somebody did was they described these repeat sequences that they found in the bacterial genome. And so that's where the term CRISPR comes from. It stands for clustered, regularly interspaced, short palindromic repeats. And it wasn't actually until 2010 that a real function was known, and Sylvain Moineau, a fellow Canadian here at the University of Laval, actually discovered that the Cas9, CRISPR-Cas9 protein acted like a pair of molecular scissors, and it actually cut the DNA inside the bacteria. And a couple of years later, Jennifer Doudna and Emmanuel Charpentier actually showed that they could take this system out of bacteria and put it in a test tube, and that they could cut any specific DNA sequence they wanted. So this was a huge advance to be able to really specifically target any sequence you want and make a change. And it was only 1 year later that Feng Zhang at the Broad Institute showed that you could actually do this inside of human cells. So we now had the capability of taking any sequence that we wanted and making changes to it to the desired change we wanted to make. So how exactly does CRISPR-Cas gene editing work? So you have a cell there, and inside you can see the chromosomes, and you have some defective sequence. So let's say this is a muscle cell, and the genetic defect that it has causes a really terrible disease like muscular dystrophy. What you need to do is introduce into the cell this guide molecule. So this is an RNA molecule, a DNA enzyme a DNA cutting enzyme, which is known as Cas9, and then also the correct DNA sequence. And so what you do is you put these three things into the cell at the same time. And how CRISPR works is that it acts sort of like the search and replace function in your word processing program. So the guide molecule searches around the DNA sequence inside your cell, and when it finds that precise match where the mutation is that people want to correct, it actually gets the Cas9 protein, this molecular scissors, to make a cut at that specific sequence. And then that allows the corrected gene to be inserted at that position. And so you've cured this genetic defect, and hopefully, we can use this for therapeutic uses. But there is one potential drawback with CRISPR-Cas9, and one specific outstanding issue is that there is the possibility of having off-target cleavage. And so once CRISPR is active in the cell, it actually stays active. So once you've made the desired gene edit, the Cas9 is still around in the cell, and there's the potential that it can make cuts at different sites where you don't want it. And so your cell can repair these cuts, these cleavage events, but there's the potential that a mutation could be introduced at the same time, and that has the potential over time to maybe lead to cancer. And we haven't been using this technology long enough to know how dangerous these kinds of off-target effects might be and long-term effects. So this is the issue that I've been looking at. So how can we stop these off-target effects from happening? How can we shut Cas9 down after we've made the desired edit in the genome? And I just want to give you an idea of how widely used CRISPR-Cas gene editing has been used. So scientists have used it to engineer crops. So, for example, they've made wheat more drought resistant and more cold resistant. It's also been used to engineer dogs. So there was a group in China that engineered beagles to have twice the amount of muscle mass that a normal beagle has. And apparently they're going to use them for racing or police work or something. So there was another group in China that has engineered micro pigs, they call them. So they've made a defect in the growth hormone in these pigs, and the pigs grow to a maximum size of 30 pounds. And these are being marketed as the new pet. You can purchase a micro pig for $1,600 US. So, sheep. So on the bottom right-hand corner there, you see sheep that have been engineered, goats, cashmere goats. So everybody loves a cashmere sweater. Cashmere is very precious. It's the undercoat that these goats grow in the winter, so there's not very much of it. And so these goats have been engineered to produce more of this cashmere undercoat so we can have more sweaters at a cheaper price. And so, Cas9 has been used for really great things. I think solving world hunger would certainly be up there with these drought-resistant crops. The micro pigs and the cashmere, not so sure. So CRISPR-Cas9 hasn't only been used in plants and animals. We've also used it in mammalian cells and human cells. And this year in North America, the first CRISPR trial in humans is set to begin. So it's at the University of Pennsylvania, and it's set to go anytime now. And the first cohort of patients are patients with very severe cancers, so sarcomas, melanomas, multiple myelomas. And what they plan to do is they're going to take the blood cells out of the patients, engineer them in a test tube, and then put the cells back in. And what they're doing is engineering the immune cells to hopefully specifically attack the cancer cells. And so they're hoping that this will be a new treatment for cancer. And this won't be the first time in the world that it's been used. Last November, there was a single patient in China who had a very similar procedure done. So we have already moved CRISPR-Cas technology and gene editing into humans. So another use that CRISPR-Cas has been proposed is for engineering mosquitoes. And so what they hope to do is engineer mosquitoes that can't transmit the parasite malaria. Now, they will do this when they engineer these mosquitoes. They actually will be carrying copies of the Cas9 and the guide, and what they will do, then they can transmit that to their offspring. And so, it's a little bit of a scary thought thinking about engineering this technology into mosquitoes that once you release them, you have absolutely no control over what happens. And so, I think when we're thinking about these kinds of technologies and engineering and releasing things, that we really need to be thinking about what are we going to do if something goes wrong. And so sort of a fantastical thing that's being done right now, George Church's lab at Harvard University is actually trying to re-engineer a woolly mammoth. So the closest living relative is the Asian elephant, and there's about 30 to 35 changes that they think they need to make the Asian elephant very woolly mammoth-like. They've made 15 of them already. And so people are doing these things, and the technology is here. We have the ability to do these things. And, I think we really need to be thinking carefully about where we go with this technology and what potentially could happen. And so I'm just going to show you a little clip of the X-Files. So while you'll understand in a minute.
How does the virus remove the ADA gene?
A process called
CRISPR-Cas9. RNA and a protein cutting genes at exact locations.
Exactly, but in this instance used as a weapon.
So how does your alien DNA protect you from this weapon?
I don't know. The alien DNA must code for something that targets the Cas9 protein and must somehow deactivate the weapon.
And so Science fiction. In fact, this is exactly what I'm doing. So we didn't find them in aliens. We found them in something that looks quite like an alien in these bacteriophages, the viruses that can infect and kill bacteria. And so I'm just going to show you a quick movie of what, oh yeah. Oh, I didn't, no, this movie has no, this movie, sorry. Phages don't talk. No, so this is a movie of what a phage looks like. So there it is. So it has its genome inside that big round head that you see, and its tail is how it attaches to the bacterial cell. And in a second here, you'll see it landing, attaching to the cell, and then it's going to come down and infect the cell. And so we found genes that can shut down the CRISPR-Cas system in phages. And so just a little bit of biology, how we discovered them. So there's the phage infecting the cell, and when the phage infects a cell, the bacteria recognizes that it's being infected. And what it does is it grabs a short piece of the phage genome and it incorporates it into what's known as the CRISPR locus. And so again, that's where the term CRISPR comes from, clustered, regularly interspaced short palindromic repeats. So the repeat sequences are the black diamonds, and between each of these repeat sequences, there's little pieces of phage genomes that have been inserted. And so this serves as a sort of molecular library of all of the enemies that this bacterial cell has battled. And so what happens is this CRISPR locus makes these little guide molecules that can recognize specific DNA sequence. It forms the complex with the DNA cutting enzyme, and this is called the surveillance complex. It moves around the bacterial cell, and it's looking for these invading viruses. And when it finds one, it binds to it, and it cuts up the genome, and that's how it protects itself. So that is actually what the CRISPR-Cas bacterial immune system is. And so phages, actually, we found, have anti-CRISPR genes that are able to inactivate this CRISPR-Cas system. So, of course, there's this evolutionary battle. Bacteria want to protect themselves from these viruses, so they evolve this immune system. But the viruses, of course, want to be able to bypass this immune system. And so they've evolved these genes that actually allow them to block the activity. So on the left-hand side, you see in a normal infection, if the bacteria is resistant to the phage, when the genome is injected, the phage are chewed up by the CRISPR system, the DNA is cleaved, and they die. Phages that have one of these anti-CRISPR genes, what happens is that a protein is produced, and it binds to that DNA cutting enzyme and totally blocks its activity, so it can no longer kill the phage. And so in collaboration with Eric Sondheimer's lab at UMass Medical School, what we showed was that you could introduce these phage anti-CRISPR genes into human cells, and we could prevent Cas9 activity from happening in human cells. So what can we do with these CRISPR-Cas off switches? So there's a number of things that we're thinking, a number of uses we're thinking of. We can use them to control where the editing is done in cell type or tissue type or organ type. So right now, with genetic engineering, when we are trying to make these changes, we use viral vectors to deliver the CRISPR-Cas components. And these viral vectors don't have very good tissue specificity. And so if you want to be editing a gene that's present in the liver, you don't want the Cas9 to be active in the heart and the lungs and the muscle. And so we can add these anti-CRISPR genes to keep the Cas9 turned off in every tissue but the one that we want it to be expressed in. So another thing that we can do is use them to control at what stage of development the CRISPR-Cas is active at. So when we're thinking about making edits to embryos, that we can control to specific developmental stages and have it turned off at other times. And of course, our gene drives. So these potential malaria-free mosquitoes, if we release these, if there happens to be a problem, we really want to have a backup in our pocket, that we want to be able to release something that can actually counteract and perhaps correct whatever is happening. And so finally, I think clearly everyone who's here is interested in CRISPR genome editing. And I think we are at the point where we can engineer the human race. And I think we need to be thinking very carefully about the kinds of things that we're willing to do. So in the scientific community, there's a lot of discussion right now around the ethics of CRISPR-Cas gene editing. And I think some cases are very simple. If we can cure cystic fibrosis and Huntington's disease and cancer, that's a very clear use that almost everybody would be in favor of. But what becomes more complicated is, where do we draw the line? So do we allow parents to choose the eye color or the hair color of their offspring? It's pretty innocuous. It's not really hurting anybody. Do we allow people to engineer their children to be incredibly brilliant, right? Do we make another Einstein? Do we allow people to engineer Olympic athletes? So I think we are really on the cusp of these things really being possible. And I think it's very important for the public to be thinking about these kinds of issues, because it shouldn't only be the scientific community who is making these decisions. These issues affect us all, and everybody needs to have input into it. And so, of course, scientific research isn't done in a vacuum. So this is the Phage group here in Toronto. You'll notice how young they all are. So almost all biomedical research in this country is done basically by almost children. They're in their 20s to early 30s. It is. It's amazing to work with them. They're an incredibly bright and talented group of people. And of course, none of this work that I do could be possible without the support of the federal government, and that's all my funding sources. Thanks.
So while we are changing computers, I think that the stream is not working. I'm going to check anyway. So I would like to introduce you to Delilah Onorato. I think you have to put it a little bit higher, because otherwise people are not going to hear. Okay. 1, 2, 3. Say something so that something. So you can hear. Okay, fantastic. Yeah.
Thank you. Thank you very much for the explanation. I wish I had access to all that information before doing my presentation. So I'm here kind of representing the non-expert. No, fully representing the non-expert. I couldn't think about a better reason for being here. I enjoy very much the monsters of this world, but CRISPR isn't exactly the subject I usually discuss. So I was trying to think about what are, the imaging that the word CRISPR actually can bring up to our minds as a citizen public that possibly tries to find the will and the strength to search for more information. So I got online and I got this image. It's a kind of advertising, so I'm in media theory, that helps. And I would like to go through appreciate a little bit the objects that are showed, and the colors used, and words. So I work in a country different than the country where I was born, so I teach in a different language. So words are like objects to me, and right now that I'm trying to speak English, it's again the same issue with words. So let's look at words like not really meaning something very meaningful, but like symbols. So when one actually reads this, like we just changed the way you edit genes, I would like to focus on the word edit. So words such as this, editing genes, sometimes are connected, and I heard you talking about mutation. So what is mutation? I don't know. I tried just to, things gone really wrong, birth of monsters, scaring change. Another word, manipulation. So what is manipulation? Bad people do it, controlling something we should not be touching in the first place. Genetically modified organisms. Okay, we know that we should not eat that, but we really do not have a way of identifying them in supermarkets, and eventually we will. Yes?
I have a quick question. I don't see any mainstream scientists saying that we should not eat the animals.
But, yes, I am. Thank you for the question. Interaction is good.
Reading to this like 2 or 3 years.
So the word editing is actually a very objective word. It's not very charged with, like the previous words. So it doesn't put us in a mind frame that is strange, or it doesn't make us suspicious about the technology or the media. So commonly applied to text, also genetics. So what is editing? To prepare, to assemble, to alter, to modify a gene. More or less this was the image we had before, but now we have the person inside. I think that many of the imaging used in medical books, they do not include the subject. I mean, you can always see, for example, a baby inside a tummy, but you rarely see the body of the woman involved. So in this case, I picked up an image that you can find again through a simple Google search that includes the scissor that we have referred before, scissors and the hand and a woman. So this, symbolically speaking, okay, when we show hands, we are automatically connecting this image to another powerful image, which is not really very scientific, but for sure very artistic. So those hands are powerful because they're connected with creation. Other hands, those are the fates from Goya. And we can see there's a scissors somehow here, right? So they're cutting the string of life, yes. Again, scissors. And connected with, well, Genesis, birth. So actually when we see the first image, like now you can edit your genes in a different way, most probably we are associating all this imaging without not being very conscious of the process, but they are there because they're part of our culture and what we have, what we have seen before learning about Crispr. So, in a very non-scientific way, I've decided to create or to give some names to the different opinions that one can have towards the medium called Crispr-Cas9. So let's say that there's this perspective, the mostly negative, which is we should not do this kind of thing. So I will call it the dystopians. Thank you. And mostly, thank you. Mostly we can connect the dystopians with a big tradition of in the film industry connected with things gone wrong again with experimentation. So one of the examples I would like to refer is The Fly, or maybe Total Recall, when economy and technology become very obvious when you slice. Yes. On the other hand, they're the very enthusiastic. So, yeah, come on, baby, like my genes. They're probably the fantasy fans. I feel that I go towards this group. So when we talk about Crispr-Cas9, most probably the first things that we would actually like to see being made are mythological creatures. Because one thing is to read about them or to see some imaging, but actually to have them moving would be really in real flesh. Because digital arts are not enough. There are cute mythological creatures, not so cute mythological creatures too, but they're very, very challenging. Okay. And your example, thank you very much, recovering extinct life forms. I call this group the guilt trippers. Yes, I'm happy that my photo is from the dodo, okay? And not the mammoth. We ate it. Okay, and then there's this group too. Stop joking, this is serious. And I call it simply the health issue supporters. I mean, we should use this technology because it's important, because we can save lives, because there's diseases that are, it should be treated, and issues that we shouldn't, we should be able not to live with it, or we are better without them. In my case, let's say, there's also the ones that go overboard. It's not just treating health issues, but actually making us better. And I think you talked about choosing the eye color of your babies. So why not being stronger? Why not, just being more intelligent? So the enhancers. I'm not sure about the image, but this was eventually this is kind of boring, because when we think about enhancing, the majority of the times we're talking about the same patterns. I thought about bringing also the modification group, and I was discussing this afternoon the subject with Marta de Menezes about maybe the possibility of having wings, but there are some problems and issues, and I'm sure I will ask you afterwards concerning balance, that we cannot actually, fly with those wings that we could have. I'm an enthusiastic towards a fantasy group. So, in 2015, when I first heard about this medium, and I read the first articles about it, believe me, my initial question was actually, after this part of the presentation, could Crispr-Cas9 make X-Men a reality? And this is not a joke. Because there are issues that are represented through X-Men that could actually be extrapolated towards society. If you remember, there's a diversity of mutations, right? And there are problems concerning social integration. So being too beautiful, being too powerful, being too ugly, it's not the same thing as blending in, right? So X-Men, well, they appear in 1963, and it's very obvious right from the beginning that there is something called a mutant phobia. They are considered a menace. So maybe, when we start using this medium in a wider way, we will have issues concerning not only medical ethics, but also how can we as a society give equal rights to genetic modified human beings. Okay, concerning my initial question, no, we cannot do X-Men because there's an X gene that we actually do not have as human beings. So my question was answered right from the beginning. So I think we should, as a society, maybe we should become more empathic, more understanding, create a space for, first of all, education, yes, and also a legal support, because most probably there will be strong issues concerning equality. Okay, we have so many already, but are we ready to receive modified human beings? Okay, and I hope there are some more questions afterwards. That was it. Thank you.
Technical support?
It should be pretty easy.
We're calling technical support. Just switching the, make space for Mark Lipton. And make sure.
That's all I needed. I just need to look pretty. Okay. Yes. Okay. So no. Let it go. I'm just going to talk, and I have pictures that you can look at, and it will cycle through a couple of times. I was compelled to use some images by my friends today, but I'm not going to speak to them precisely. I'm going to speak to you. I wanted to begin with, I have three things I really want to say. But first, I want to express my gratitude and thank you for the invitation to speak here. What an honor, and thanks to all of our sponsors. And thank you to the researchers and the scientists who are doing this work, and the other scholars and people who are raising these questions around ethics. I have some questions about ethics, similarly to Dahlia, that I want to ask, and I think we had one overlapping slide, but if you ask two media scholars to talk on the same topic, it's going to happen, I guess. Just one, yeah, just one. I'm here to really promote an appropriation or of CRISPR in a way. I want to celebrate the biohackers of the world. And I have 10 quick examples that I want to share with you. And then really I sort of tell you where my point of view comes from, and sort of raise a final question about how we might all work together as time goes on. But I wanted to introduce you first to the DIY bacterial gene engineering CRISPR kit for $159. There were 2 pictures of the kit. It comes with the example experiment that teaches you many molecular biology and gene engineering techniques. I can't guarantee that to you. I can tell you that CRISPR-Cas9 is so easy, even middle schoolers can use it. In middle school, at least in Ontario, we do something called a science fair. CRISPR-Cas9 is one of the most popular science fair experiments currently being implemented by students. And in a way, I think it's great. I think that we're getting people excited about what the possibilities are in a very tangible and a real way. I think when not just your students who are young, but these middle school, how old are you in middle school? Like 12, 13 years old. So we have to begin to think about, I think, a lot of us are scrambling, not just thinking about rules for, the biological impacts, containment, introduction, and disposal of whatever materials it is that we're working with. I like the example of the startup Perfect Day, where they've used CRISPR to make milk and dairy products without lactose. I don't like lactose myself, and neither does my family, in that they don't like me to have it. But it's a 3D printing, yes, thank you. It's 3D printing of the DNA sequence of a cow, and it's inserted to a specific location of yeast. They were able to get this DNA sequencing of a cow, which they named Buttercup over at Perfect Day, and they've obtained it from the U.S. Department of Agriculture, right? So it's yeah. I mean, and the Impossible Burger, there's a picture of a couple of hamburgers there. I can't guarantee that Impossible Burger is made with CRISPR-9. I'm not completely sure about that. But I do know that it is a non-meat burger where the ingredients include wheat protein, coconut oil, protein powder, and heme. Did I say that right? Heme? Have you heard of heme? I haven't heard of heme either. Heme is responsible for the characteristic taste and aroma and the bloody mess that comes with an Impossible Burger meat. It catalyzes the flavor when the meat is cooked. Heme is exceptionally abundant in animal muscle, and it's a basic building block of life in all organisms, including plants. Impossible Burger discovered how to take heme from plants and produce it using fermentation. And when I start hearing reproducing and fermentation, CRISPR can't be far behind. That's sort of why I started there. I recall in the U.S. the National Institute of Health refusing to fund research into gene editing technologies in human embryos. And the arguments, I think, as Delilah pointed out, against human genetic engineering are mostly based on social stigma, fear of the unknown, religious ideology, and anti-science rhetoric. And I'm not here to give you any of that. I'm here to talk about biohacking as it brings up concerns about ethical responsibility, along with the fears that terrorists will exploit the open source methods associated with CRISPR. And while posing these important ethical dilemmas, the ethos of biohacking also suggests new ways of thinking about the body, health, medicine, as well as aesthetics and other philosophical issues. And I am drawn to call for more international and independent biohackers to play with and create more bioart. I give the example of the Critical Art Ensemble's contestational biology project that seeks to challenge prevailing bio-related industry norms. They resist dominant ways of looking at science, the Critical Art Ensemble. They resist things like biohacking as creating a new version. They created a new version of Monsanto's herbicide-resistant Roundup-ready seeds. Our knowledge of biological systems are becoming increasingly more complex, and bioart signals possibilities of future innovations for both creation and production and an ethics of resistance. And so by questioning basic assumptions of commercial biotech, various forms of bioart are creating new meanings about intellectual property, biology, and life. So somewhere in there is one of my favorite bioartists, Natalie Jenko. I hope I got that right, Jenko. Natalie is at New York University, and she's the author with Eugene Thacker of the Creative Biotechnology, a user's manual. It's available online as a PDF. This is not a guide for dummies. This is a book with explicit instructions for executing basic genetic procedures with minimum equipment. A couple of hacks that are sketched out include cloning a tree, starting a culture of your own skin. Yay, I think that's great. The second biohacker that I show up there is Josiah Zahner. He's the one with the white in his hair, like me. Josiah recently injected himself with the Crispr, the powerful gene editing, to hack his own DNA. So what's going to happen to Josiah? We don't know. And I think that the self-injecting, it's not a new thing, but it is an ongoing ethical challenge. Smallpox began when a scientist injected himself. And similarly, polio, the inventor, injected himself, and then for some reason both of those turned to children as injection participants in their research, which was not ethically endorsed. And so I raised the issue that middle school students are now having access to CRISPR technology. Is it right or wrong if you inject yourself? I can't tell you what to do with your body. However, I do think that if it's in the name of science, the ethical questions are raised very firmly. If it's in the name of art, they are not. So that's something I'm keenly focused on and interested in. And it is Josiah Zahner who's selling the DIY gene editing kits. Way to go. The picture of the hand with the light on it is not a new idea, and it is not CRISPR. It's part of grindhouse wetware. And I've read a few years ago applications for government funding to study the grinder community. Now, like, I'm thinking grinders, like the meeting gay boys online, but that's not what this grinder is. These grinders literally are biohacking or using surgery to augment humanity. So a magnet in your finger that's implanted into your finger will help you open a door or pay instead of having a credit card. That kind of technology really isn't that far away from what CRISPR is. And I think it's still really interesting to sort of put CRISPR and the grinder community next to each other. The grinder community is mostly underground, whereas CRISPR is mostly real scientists solving real problems. Then just a few shout-outs to some of my favorite biohackers around the world. In New York, Sebastian Cocabio, I hope I got that right. He works on plant engineering and photosynthesis. He also works with local biohacker spaces in Brooklyn and at the Visual Arts School teaching plant engineering. In Brazil, in Sao Paulo, Gypsy Lab 8, yes. They tinker with all sorts of stuff, from bio to electro sensors. And in Brazil in particular, there is this worldview that is around DIY and improvisation. They're already deeply embedded into the electronic, bio, and digital practices and philosophical approaches of tinkerers, makers, the practice of impromptu makeshift, resorting to quirky, smart improvisations. So I think the biohackers in Brazil are doing something that comes very naturally. In Switzerland, Marc Dusselier from Hacketeria, specifically in, all these, you're laughing at me, in Zurich and Lausanne. He travels with his own lab equipment to teach critical design. In fact, he's been doing biohacking before the word biohacking was really part of our common usage. And I think he calls himself an interdisciplinary maker-educator. And then I just have 3 more quick examples to share with you. Excuse me. In Chile, Santiago, the Backyard Brains, which is now 5 years old. I don't know if you've heard of these groups. You can find them all pretty easily online or on Twitter or whatever social media you are in love with. If you are, my students are, so they would be looking at it. Anyway, the Backyard Brains is dedicated to microscopia, and they're famous for their wired cockroaches. I'll say that again. The cockroaches. They biohacked cockroaches. Okay, in Mexico, Interspecifics is an intersection between art and science, and they embrace hybridized practices across disciplines and living organisms. They're also really interested in the relationship between open knowledge and the precarity of their work, right? Like in Canada, we're fortunate that there are governing bodies that fund medical research, fewer for bioart or biohackers, but there are ways to work within systems in Mexico, not so much. And finally, in Vancouver, Canadian, the Maker Mobile Tech Learning Lab, and this is the truck that drives around the under-resourced communities in Vancouver, where curious kids can have a look at the lab, see what a lab actually looks like, and they can learn to repair their broken electronics, among other things. So those are my 10 shout-outs to my favorite biohackers or biohacker critics. I sort of come to this really not as a biohacker and not as a scientist. I think of myself as an educator. The educator is really how I identify myself, and I take a philosophical approach that I call a media ecological view of the world. So we can see like what on this earth is ecological, and everything that isn't would be media ecological. And the relationships among those human-made things with the ecological world and those ongoing systems and interrelationships are what I'm particularly interested in. Matthew Fuller has described media ecology as a dynamic interrelationship of objects and human communication processes, for my fellow communication scholar. I think of it more as sets of integrated paradigms. So I can now steal all of Delilah's science fiction, and I'm going to steal what I can't, not steal. Stealing is bad. I cite. No, I think a better word is cannibalize. I'm a cannibal, really. If it interests me, it becomes part of my worldview. And so when I think about the biological sciences and biogenetics, I actually fall immediately into this place that I'm not comfortable in. And that's the sort of basic binary dichotomy between nature and nurture. I prefer to see nature and nurture as important and valuable and working together all the time, among other things. I'm a both and person. But when I first looked at the technology, I went back to the either or. Because I look at our society as a whole, and I think we have an invisible public health issue that has been referred to as child abuse, childhood trauma, or developmental trauma. In fact, scientists have failed to find consistent genetic patterns for mental health challenges like schizophrenia or bipolar. There may be genetic factors that predispose people to develop trauma or traumatic stress. I'm not denying any of that. But and I guess what I'm saying is I don't see genes as fixed. I think that life events in and of themselves can trigger biochemical messages that may turn certain genes on or off. This scientifically, and you can please correct me if I'm wrong, Karen, attaching methyl groups, a cluster of carbon and hydrogen atoms to the outside of a gene, the process of methylation, making the gene more or less sensitive to messages to and from the body. And then I think about these sort of traumatic issues and the role of psychotropic or psychopharmaceuticals. And you know, like I love my psychopharmaceuticals as much as my students, but they really can only do so much. How can we ensure that humans can become, like all humans, can become contributing members of our world? Because in fact, methylation patterns, as I understand it, can be passed on to offspring in what is called epigenetics. So how can we make sense of human suffering today? CRISPR outcomes definitely can relieve human suffering. There's no question about that, present and future. But I would say all media change is a Faustian bargain. Some win and some lose. And I'll just end with this final thought, because I mentioned funding models and funding bodies. In Canada and around the world, ultimately I'm calling for more multidisciplinary teams working together, taking that longer view and solving current human challenges together. Researchers across the sciences, I urge you to include the artist, the tinkerer, the maker, the creator, the player, the ad hocer, as well as educators like me, to help consider our human challenges in the years, the decades, and the centuries ahead. Thank you.
It's my own computer. This is why I'm the taking it. My computer was collaborating today. Sure. No.
You need technical support.
Yes, it just stopped and yeah. Yes, I really don't want to. It's just I can't find the mouse. Okay, so let me just. Yeah, it doesn't do that. It does. Okay. I'm sorry, I have to restart.
That's fine.
It's.
Say no.
That image. Don't worry, it's been a couple days with computer issues, so it's not just your. Come on. But the talk is here.
Oh, that's good. It's not going to stick or anything. Okay.
It's not.
So okay.
That's not going to help.
A little bit of patience.
Sorry.
It won't take a while.
It's coming. You can go to the toilet quickly. You can come back. The mathematicians drunk it all.
I think people are rushing. Yeah, because usually during the day.
Okay.
We arrive before the school.
This looks better.
Yeah, I think it's gonna.
It's okay.
There you go. Woohoo!
I like straight
Some people rush to the bathroom actually, so like.
Go away.
There really is no. I did not hack your computer.
You didn't. No, it's a little old computer. I need a new one. It's coming. It's coming. This is not the one. Okay. Hopefully this will. Oh no, that's a very bad. Okay. Okay.
Okay, so we're starting. Non-dimension.
Oh no. So for some reason I'm getting claps just even before I start talking. Thank you for all the support with the technical support. So I'll try to be brief. It's a little bit complicated. As you can imagine, talking for 15 minutes is not, it's a challenge. For most people, I find it very challenging to actually summarize the things that I do in 15 minutes. It's a little bit complicated. The first slide that I'm giving you is actually not from art. It's from design thinking. But I do like it because I like to explain that for every work that I do as an artist, which is what I am, I start by empathizing with the subject that I'm going to be working with. And I think this is really important. Okay, so I empathize. I try to research all the knowledge that I can in different fields, in as many different fields as I can access, about the idea that I'm looking at, the subject that I'm empathizing with. Then I go into trying to come up with a form to give the idea. So I am an artist, so I'm a visual artist, not just another kind of artist. But as a visual artist, I'm trained to put concepts into shapes. Okay, this is what I do. So in some part of the process of creating an artwork, I need to imagine what it's going to be. And for that, basically what I do is I decide what is the material that I want to use and what is the technique that I need to use to make it into something that I want, to express the question that I'm putting forward, to push forward the subject, the concept, whatever it is interesting me. And then I do prototypes, and then I do experiments, and then I test the technique. I test the material. I see if it works. I make decisions through the whole process to come up with a piece that then at the very end of the process I can present to you. I can present in an exhibition, and you can look at it and tell me something that I don't expect. And sometimes I have to go back and redo it. But with experience, I manage to actually come up with things that I don't actually see as mistakes. So this basically, for the last 20 years, allowed me to go into many different kinds of research laboratories around the world, because basically what I do is I manipulate a living material, whether it's a whole organism or a part of it. And by doing this, I express a specific question. So just to give you an idea, I've been in evolutionary biology laboratories. I've worked in tissue culture. I've worked with proteomics. I have worked with microbiology, plant biology. Microbiology, medical imaging, cancer cells, HeLa cells, muscle cells, different kinds of materials. And I'm here really to tell you about my latest projects and the works that I'm doing, because I have, over the last 2 years, been thinking about projects that use CRISPR as the technology to manipulate the material. And one of them is called the origin of species. This is a project that I'm doing with a friend, a philosopher, and it's the first time that I'm working as a co-author with a philosopher. I've worked with scientists a lot. This is the first time that I'm working with a philosopher. She's called Maria Antonia González Valerio. She's a Mexican artist, a philosopher, sorry. She's also doing art. And in all of the works that I did so far, there was very little genetic manipulation. So actually,
a lot of people look at my works, and because I say I manipulate a living material, they automatically assume that I'm doing a genetic modification. And I have to explain, actually, there's a lot of ways that you can change a living material without resorting to genetic manipulation. It has only been, when CRISPR came about was the first time that I actually wanted to do projects that use genetic manipulation to ask similar questions that I did with the previous works. But this is when the technology actually was right to manipulate the materials that I want to use and to make sense in terms of asking questions that I think are really, really important. And one of them relates to origin. So one of my major interests, or my umbrella interest, is issues of identity, whatever perspective you want to bring to it. If I want to think about myself as a human, if I want to think of myself as an animal, as a woman, as a mother, as a daughter, as an artist, as a researcher, as Portuguese, as blonde, or, doesn't matter. Whatever you can think of is a new perspective on identity. So whether I want to include my brain function in a portrait, or if I want to make a protein that has my full name as its amino acid sequence, I'm always wondering, how can I enrich the idea of identity? What is technology and today's society bringing into the plate for me to understand who I am? And when I understand who I am, I can position myself in the world. I can make decisions. I can decide to support no GMO or eat GMO because I don't believe it's harmful. Okay, so these are the kind of questions that I'm looking at. And the origin of species is about the origin. So instead of trying to create monsters that are in fantasy or actually very much embedded in our culture, I thought, can I go backwards in time? Can I actually, instead of looking for inventing genes that I have no reference for, can I try and find genes that existed before? And does that make a change? Can I look for the origin of the genes that I have today? And this doesn't necessarily, we'll talk about it later. So this was the premise for me looking to CRISPR to actually see if I can make this a reality, and what form does it take? What are the biological constraints of this technique, and how far can I go? And does it make sense to go any far? What makes sense in something like this? This piece was actually inspired by the work of George Gessert. George Gessert is an artist who lives in Oregon. He has used living material for his artworks for a very long time, but he does, basically he breeds plants. So he crossbreeds them in the very traditional way of plant breeders. And he has this idea, or this project, that is very beautiful for me, which is the unattainable memory, an image of a wild iris that he remembers encountering in a field when he was young. So he knows that the memory is tainted by all of the emotion that he had during that day, whatever those were. He knows that the memory is tainted by the time that it has been between now and then. And so he knows that it is unattainable. This iris is unattainable. He's not going to get to ever see it again, but he still endeavors to find it for different reasons, through different strategies. And I really love this impossibility in a way. I think also this is what brought me to the origin of species, which is something that I think, it's impossible. You cannot reverse time. It doesn't really make sense in this context to say that you're recovering genes, because you don't know if the genes that you're assuming are the original genes are actually the original genes. You can't compare. So what is this thing about this impossible endeavor to go backwards in time and recover something, and why is it for? The other project that was very inspiring for this piece, and that brought me to Maria Antonia, was the piece by Bios Ex Machina, where they grew corn in Mexico City that came from different origins and animal feed as a device to see if there was genetically modified corn in Mexico. So it's not allowed, it's illegal to have genetically modified corn in Mexico. Mexico is very, very protective of corn in all of its varieties. It's part of their national identity. And so this piece basically grew the corn. They sprayed it with Roundup every week. And if there was a plant that survived the Roundup pulverization, then they would take that to the scientists to see if it was a spontaneous mutation or if it was actually a result of hybridization between across the border between Mexico and the United States. So when I spoke to Maria Antonia, we decided, okay, so can we do plants? Can we do corn that will be an ancestor for corn? There's a lot of variety of corns in Mexico. There's a lot of loss through policies like monoculture happening. Can we recover some of those species? Does it make sense to recover some of those strains of corn? And what are we actually doing with such an objective? So one of the interesting things that you need to think about is, even if you can, with the genes from here, even if you can find what were the genes of the plant that was before the branching happening, you cannot compare to it. We don't have that plant anymore. And actually, you're not going backwards. You're just adding a branch to the tree. So that impossibility of going backwards in time is something that I'm really interested in. That no matter how much you try, what kind of strategies, very reasonable strategies you may devise, this is not really happening. And you need to understand how it works. Curiously, it's completely impossible for us to do this project in Mexico because talking about genetically modified corn, doesn't matter for what objective, in Mexico it is impossible, okay? The scientists would really get their funds taken away if they embarked in a project like this. Not all over the world. So in Cuba, actually, we found a laboratory that is working on this. And we've already made contact, and hopefully we will next year do a residency to see what are the constraints. How do you find a gene that was there before? How do you make an educated guess of what it looked like? Okay, because you're not actually finding it. You're just making an educated guess of what it was. Okay? And that's the practical part of it. So the corn is the material, the CRISPR technology and all of the analysis and archaeology, genetic archaeology that we're going to be doing is just technology. So what is the meaning of a piece like this? What does it bring forward? It questions the idea of an identity of a nation. How can an identity of a country be so connected to a plant? You know, they eat it. They eat it every day. They eat all of the varieties that they can. If you're there, they will feed you the corn in whatever form you can think of. We outside of Mexico basically only eat yellow sweet corn, right? They have a multiplicity of corn, and it is, I don't know about you, but as a Portuguese, when I go into another country, the first thing I go and do is eat local, right? Because this is part of what I want to learn about a different culture, right? Because it's part of their identity. So if food is such a big part of who we are, I come here and I eat maple syrup like there's no tomorrow because I don't come here too often. So it's see, I don't go to a Chinese restaurant in Canada. Really? And maize is not just their food. So corn is also a political statement. The fact that they do decide to eat corn is a statement of their identity. This is who we want to be. This is what we are. This is who we were. And the, colonization cannot take this away from us. Whatever colonization it is, if it's economical, if it's political, if it's historical, they cannot take this away from us. So it's a statement. The second project that I want to talk to you about that I'm working Crispr with is this one. So I am going to give you a question, and you are very free to interrupt and actually give me an answer. Perfect. So which of these mice is more natural for you? None of them are dead. Yes? The hairless one? Okay. Did you answer that because I asked the question? and you thought that I was, tricking you? Anybody has a different answer? Okay, so just the fact that I asked the question is conditioning your response, okay? If I ask the question, it means that maybe I'm tricking you, and actually the one that doesn't look very natural for you is the one that is more natural, or maybe you actually think that there's no such, it's really very unusual to have such white mice, and that cannot be natural. So the question that I want to raise with this project is actually, what makes you decide if something is natural or not? Okay? And in this particular case, these mice were they were something that I suddenly found as being, okay, this is the best material I could possibly get to ask this question. And I will try to explain why. So the white mouse is actually a really common laboratory mouse. It's called bulb C. It's in every scientific research that you work with mice, you will have bulb C. It was, as a strain, it was popularized in science from the 1920s. And then the left mouse, which doesn't have any hair, is actually a spontaneous mutation of bulb C. And I repeat, it is a spontaneous mutation. It was not engineered to be like that. It's a spontaneous mutation in only one gene, which is also really important. It's only one gene, and the effects are, it doesn't have any hair, as you can see, and automatically you go, oh, poor little thing. But more importantly, it doesn't have a thymus. And this means that it is severely immunodeficient. Immune mice are really good tools to have in a laboratory, especially if you're studying the immune system. Okay, they don't have an immune system. So interestingly, if a mutation like this happens in the wild, those animals are not going to survive because they don't have a robust immune system to deal with whatever infections and pathogens that they come across. So it was very fortunate that it happened in the lab and that they could keep the strain going. And now it's actually a really expensive and important tool for immunologists and other scientists around the world. So again, I asked the question, which one for you is natural? I would argue that either one is, but it is interesting for you to think about what are the criteria that you're using to decide on something like this. And I think this is really important because when we start thinking about manipulating humans, you need to think about what are the criteria that you're using to make a judgment. Okay, because basically you're making a judgment. Because calling something unnatural is a negative judgment. It doesn't have to be, and maybe we need to rethink about this, but it is, until now, a negative judgment. Okay, so what am I proposing to do with these mouse? Would you endorse me in trying to fix it? And what do I mean by fixing it? I'm fixing one gene. Through Crispr, it wouldn't be very complicated to do this. I could do it at the germline, and I could do it to an individual, but I would be using an animal for an art piece. And that is the first ethical challenge that you would face. But also, you can clearly see that this gene being mutated is something that hinders the mouse. It cannot go free. It needs to live in a bubble all of its life. They need to sterilize the food. Actually, this is a mutation that occurs in humans as well. You have humans that have this. They're hairless, and they also have a very severe immunodeficiency. So this is the FOXN1 is the gene that was mutated. And so this is something that hinders them. But what if it's just about the hair? Would you accept that we change the mouse just to make it recover the hair? Or is it really that we're saying that, okay, it's going to give it a better life, so we should do it because it will give the mouse an immune system back? But, when you change one gene, you can have multiple effects. And how many of those are a matter of enhancement or a matter of curing? And what are we curing? How many things do we call diseases that may not actually be diseases? Maybe we need to think about what a disease is and how does it actually change our life or affect our lives. And are we killing variety just by deciding that we all want to have, every man that I know that has baldness wants to not have it. Are we just never going to have bald men ever again? I would miss them. Okay, I'm married to one. I like him. So this is how I will present this, and this is my last slide. So please engage in asking these questions. I think all of the speakers tonight were, very forward in bringing you challenges, ethical challenges, intellectual challenges for you to think about. So thank you very much for listening. And I want to personally thank all of the scientists and colleagues that worked with me on all of my projects. So this is it.
I don't need a microphone anymore because the room is this is what they told me.
What do you mean the room is microphoned?
So, but if you I got a microphone for the audience who's hearing now. I would suggest that your volume of voice higher. But you all have quite a nice voice. That's good. Nobody is whispering here. But I would like to open the floor to questions.
Down at the level of microorganisms, there's huge amounts of gene transfer going on naturally. And if you were a bacterium, it would not be safe for you to have your genome messed with all the time. by Phages and by others. But up here at our level of the biosphere, we tend to think species are really important, and our genomes are some kind of legacy of generations of slow evolution. But in 1,000 years, we may be like the microorganisms. And so biology, so this moment may be the moment when evolution on Earth, for large creatures like us, becomes more like what it is already for microorganisms, where nobody has a genome that's constant, your children aren't like you at all. Of course, in your life, you may completely modify the number of arms and legs you have, or whatever you want to do. And everything, the whole biosphere will do this. Is this rocket for you?
You've been on second line?
The gene is under the law, and if high school kids can do this, everybody can do it, as much as they can.
Actually, Mark has a good point. If you ever been in such a life, it already happens, just not in the so-called real world, but use the virtual world real or virtual.
Wishful.
Yeah, wishful. So.
I don't know whether this is a good thing or not, but it seems to me inevitable that the fundamental basis of evolution will change for everybody.
I'm not sure, but I don't think we're that far.
I'm sorry, but it doesn't look like it's microphoned. It's not present.
Okay, so heels, we're gonna use the microphone. Sorry.
I'll get it.
Okay. Just use it.
Do you want to take it?
No,
Or we just...
the people who ask questions don't need the microphone.
Okay.
And
You can repeat it? You can repeat the question?
If you can repeat the question. I cannot. I cannot.
Okay.
Maybe evolution is different from now on, because we're all going to be like microorganisms.
There's people with different listening abilities.
We're all going to be like microorganisms who are comfortable with constant gene manipulation and change. And it won't be just people who do this, but the whole biosphere will do this eventually. That's what I think.
I would suggest that
Farewell, species.
So I was just saying that I don't think we're there yet. I'm not sure if we're going that way, in the sense that actually we may be going in the opposite way. We may be more protective of our genes than we ever were before, because we will know how to also decide that. So it's not just that we may decide to change everything, but we may decide to preserve things. I would just say we're not bacteria. So, when you look at human genomes and chimpanzee genomes, it's like 99% identical, right? So the differences between us and what are arguably very different life forms than us are very minuscule. Like, I think we share 70% of our genome with yeast. So I'm not sure that answers your question, but I'll pass it along.
You want me? I was just going to refer to a mathematician that I've cannibalized. Was that the word I used? Norbert Wiener wrote in The Human Uses of Human Beings about the differences between a medicinal use of strychnine and a deadly use of strychnine is really only a matter of degree. And I think what he was talking about then was like how the information environment had changed so radically that change itself has changed. And what you're calling for is more than a matter of degree, but is something that's radical. I at the same time, I support and promote that kind of radical thinking. I recall saying to my class yesterday, there's no such thing as a human body. You don't really have a body. That's a reification using language of what is just a pattern of activity. But I do think that certain patterns of activity are more stable than others. And yeah.
Yes, I guess I just wanted to build on that question and reframe it and ask you to comment again, if you don't mind. Because I think that what we're on the cusp of, at least the language that I use, is volitional evolution. And that is different. That would be, I've decided I'm going to make this synthetic gene because I think it's going to do X, Y, and Z, and I want to incorporate that into my genome for whatever reason. And I think it's such a great one that then I'm going to go back and say, well, I'll now manipulate my children so that their offspring will have it, because it's, from my point of view, evolutionarily aesthetically pleasing, or it's interesting, or it's empowering, or whatever value I've chosen to contribute to that. And I think that that's actually different from bacteria, because I don't know that bacteria have the volitional piece. And I think that's what's potentially going to be interesting, is that we'll value something out there in the world and decide that we'll take that and incorporate it into ourselves. And I think it might be 1,000 years, but it might be less. So I'm just trying to sort of go at the same question, but from a different point of view.
Can I just want to restate that, well, if you wanted to do that to yourself, I would say, okay, go right ahead. If you wanted to do that to your children or to any children, I would say, I think there is an ethical challenge for me at this point in my life. However, whatever you do to your children, you still could not, I mean, as a volitional mother, for example, I would say you're probably like the best mother in terms of you're abusing your children. What in other ways are you abusing your children, and in what kinds of ways would that have an impact on their brains and their behavior, their sense of identity and sense of self? You can try to control their eyes, but trying to control their eyes would mean you're trying to control many other things of which you will not know the consequences. So there are unintended consequences always. There are winners and there are losers. And you think that you would be promoting winners, but you still wouldn't know. Even if it's 1,000 years away, you still wouldn't know if you were making loser children or winner children, not to say that that even exists, because I don't believe that. I think all children are amazing just the way they are. So
And we do that kind of thing already environmentally, right? So I'm rich and I'm sending my kids to the Juilliard School of Music because I have these ideas about music and mathematics, and I've decided that if they can be a math genius in the world, they'll be better off. So, I mean, parents already do this kind of manipulation. You just don't do it. But my point is, but I think that feeds off of your point about it. We are doing it, so why won't there be a few crazy people out there in the world that say, I can just do better?
I don't think it's a few crazy people. We're already doing it. And there's an epidemic of childhood trauma, of child abuse, and developmental trauma. That is my point. And so all of those parents, like, I would say, slow down. The Rumi says you go slowly making a life.
What if it was something that made you immune to a certain disease that we haven't found yet or hasn't attacked us yet? We do vaccination for our kids. Wouldn't we all do that? And then, the effect could have there could be some effect that we don't see in the beginning, but we do it and we will do it, because we see those are our children, we want to protect them. And we do it now. I agree with her, there are many things that we do right now that aren't necessarily negative right now, may have consequences in the future, and we just do that. And we're going to continue doing that.
Can I just bring back the idea of volition? And I think this is one of the challenges that I see in this kind of technology, is the volition. Is it about choice, really, or is it about complying? So I actually think that a lot of the decisions that are being made are for exactly what you say. And this is what is giving me pause, is the idea that actually decision makers, exactly like you say, that decide everybody, it's not that I don't agree with it, okay? I vaccinate my kids. But if suddenly they could reduce the probability that a child will get Alzheimer's, everybody would, or it could be a policy, just like a vaccination, to do it to every children. And it would not be about volition. It's about the government deciding for you what your kids are going to be like. and things like this. And this is actually something that I see happening because policymakers will be making those kinds of decisions. And when they do, you don't have a lot of options. You can rebel and create a society parallel, or you can just do it. And there will be advantages to one and disadvantages to that, and advantages to the other and disadvantages to the other. This is when you're reduced to a yes or no kind of decision. This is where I see the problem happening. Do I comply or not comply? And if I'm complying, what am I giving? What am I bringing to my kids? If I'm not complying, I'm also bringing to my kids a lot of issues. If I decide to change their DNA so that they will never become obese, because I don't want them to be bullied.
We have can we all raise their hands without jumping in to talk? So we have a lineup of people who want to talk. So there's first this person, and then this person here, and then another person there.
Thank you very much for your time. Can we get a better idea of the state of the art of the technology? So CRISPR Cas9 gives us this mechanisms for editing, but how hard is it to design the guide molecule, to design the replacement of DNA? Isn't that, it sounds to me like that's the hardest part.
No, that's trivial. That's the easiest part. There are programs online. You put in the edit that you want, the gene that you want to change the sequence of, and there are programs online that will design the primer for you and send you the RNA and the DNA to do it.
The mechanism to create, but how do you decide on the sequence?
Oh, how do you decide on it? So that is more difficult. So, and it comes back to a point that you made about mental health, schizophrenia. So there, right now, some genes are some diseases are, it's very easy to tell the mutations that we need to correct. So cystic fibrosis, the majority of patients have a single base pair change in the DNA, and it's very well characterized. We know what the mutations are. And if we correct that mutation, we know that we will correct the disease. But lots of other diseases, many cancers, for example, are actually multifactorial. And I would say most human diseases are not single gene defects. They're multigenic. And so it does become very difficult to try and figure out what are the changes that need to be made. And again, epigenetics is another factor. So it's been really well established now that if a mother is exposed to a lot of stress when she's pregnant, that there are permanent changes made, these epigenetic changes to the DNA of her offspring. And CRISPR-Cas is not going to solve that kind of problem.
I just wonder if we focus on gene editing as our healthcare and our culture, if we're kind of negating the need to have a biopsychosocial model of the health that also looks at quality of life that might have impacted that expression of the gene, or even an anti-oppressive lens when we look at health instead of just fixing a gene issue. Are we going to somehow feel that we don't have to look at health with a more complex lens and how those balances help people? I mean, the age study that Mark talked about, the active childhood study at hybrid permanente, really distinctly showed cause and effect of a certain age score. So I just feel like we could start losing our focus on a broader health model if people are focused on gene editing as a cure.
This is what I mean with deciding what is the disease and not. This is exactly what I think.
Or is it diversity?
So I think a good example for that is in the deaf community. So we can now give cochlear implants to deaf children, and they can then hear. And there has been a lot of controversy in the community. Should we fix this, right? This deafness is a natural condition for them. And is it right to say no, you shouldn't be deaf, and we're going to force this on you. And I mean, parents are making those decisions for their children. I actually have a friend who had a deaf child who had cochlear implants, and it was a really hard decision for her. So I think we're already grappling with those kinds of issues, but certainly I think there are potential problems, as I said, with Cas9 genome editing, that, we don't know the long-term effects of correcting genes, of making these kinds of edits. And so I think it's very important that we not be using this technology cavalierly, that we're not using it in place of proper nutrition and, not smoking and good parenting. Yeah. Yeah.
Jackie, and then another person behind, and then Susanna.
I was wanting to go on the trauma and
Can you speak up?
Okay. Trauma editing and policy in gene editing, because who gets to live by highway? It's the pollutants that actually in the air, that air pollutants that can cause cancer, illnesses, and there's policies actually that are not indirect, but direct. But indirect is who gets to live where, who gets to be exposed to what kind of air pollution, and who gets to be exposed. So that's actually a way. I think if you talk about evolution, that is something that has been changing a lot, and it actually is changing our genes. They're looking at me very confused.
No, I agree with you. It makes me think of one of the things that Roberta started with, and the land that we're on, and our First Nations community, and I pay attention. And the trauma that we, through policy and through, have inflicted on a people or a group of peoples, and I find it all very. It's scary. I want to be optimistic. I want to see the value, and like the. If I could cure myself, for example, of, all of the actual diseases and then maybe the made up mental diseases or what have you, by doing it to myself, I would do it, However, I do think that, with every new media change comes a whole new philosophy, a whole new way of seeing the world. It's like when I said winners or losers, I think it's very complicated. It's not just like, okay, well, we should all be fortunate that we're here in Canada, because otherwise we would all be in one of those, I'm not going to quote Donald Trump, but one of those other countries. And so a great deal of it, I guess what I'm saying is luck or chance, and the connection between luck and chance and risk. And with risk comes decision making, I think. So, I feel so lucky, and I feel like the chance that I've been given leads me to make certain decisions about various risks that I'm willing or not willing to make. Not everyone has the same luck or the same sense of chance. I'm trying, yeah.
We're definitely talking about first world problems here. That if we had clean drinking water and had vaccines that we could give to children around the world to prevent the diseases and childhood deaths, that would be solving 60-80% of the problems in the world.
And by problems, I mean human suffering.
Human suffering,
like,
yes.
I want to be clear on that hunger itself is not a problem. The suffering that results from hunger is the problem. And that we can.
One, 2, and 3. and 4. And then I think we should, with this gentleman here, we should pose these 4 questions in like 5 minutes. Are going to be hard to answer.
So I'll just stand and project. Thank you for the time and for your perspective and knowledge. I like the X-Men example because it was a usefully extreme distillation of, to me, an important ethical quandary, which is how do we encourage acceptance of eventual transgenic individuals? And the foil to that is, well, we have ethical problems that we can't solve right now. So what is the role of people who are at the forefront of this movement to actually accelerate the useful resolution, the constructive resolution of ethical quandaries everywhere, which will solve the ethical quandary of transgenics? Like, what role do the scientists and the thinkers have to play in that larger problem?
Thanks, man.
It's your superpower.
Yeah. Okay, I think Mark referred to a good way of getting more acquainted with the super powers and the possibilities through the biohacking society. What do you call this? Group culture. Yes. It's interesting because it's in this model of open education. Okay, that anyone that is actually interested in can have access. So it's a sort of mediated knowledge that is not connected with big science or the academic world. And there's some ethics there, despite the fact that there is a huge, I would call, negative undermining of the deontological consciousness of biohackers right now. So that would be the key right now. And if I think about anything else after the scientist, I'll let you know. My superpowers are not activated at this point. Thank you.
I don't have anything. Do you have anything to add? Yeah.
Do I have anything to add? I don't. I think also Mark suggested that multidisciplinary teams would be a really important effort towards coming up with ways of dealing with potential ethical issues that will rise from all the technologies that we're developing. It doesn't limit itself to CRISPR, I have to say. I think that's just a strategy that we should be thinking more and more and more. I think we live in a society that progressively developed towards specialized fields of knowledge, and we still are very, very not comfortable with people that have an umbrella kind of view over things because they're not knowledgeable enough, and so we tend to not be very accepting of what kind of insights they may bring. But I actually think that you need all kinds of approaches and strategies. You need the umbrella people and you need the specialists as well. And actually the umbrella people should be actually teams of specialists that could actually communicate with each other, which is something that is a little bit of a challenge, I understand. But there's more and more activity happening. I also think that those teams need to have people like biohackers involved and people outside of regular knowledge pursuing or knowledge owning fields. So in terms of traditional academia has been the entity that holds knowledge, and I think we need to think of other strategies and other forms and models for, knowledge is doesn't only belong in academia. It belongs in the regular Joe or Jane that is crossing the street. And so we need to have better access and communication between those two spheres. It's this bubble that we live in, this privileged bubble that we live in here in this room, also needs to communicate better with the people that don't come in, that they don't know this is happening, that it's a real challenge, but I think we need to get better at it.
Suzanne, okay, so just a second. It's 8 o'clock, and we still have 3 speakers, 3 people who are asking questions, and I hope that we can do the 3 of them together in the next 5 minutes.
I'm going to make sure, because in part, the last question was where I was headed, as well as your answers there. But I'm just going to interject a couple of ideas. One is, I'm wondering how we're going to get those questions into the right hands, to have those active conversations with researchers actively doing at this point, in terms of not only productive technology and other areas of practice. As human beings, we've been doing this for a very long time in various times. 20 years ago, I was on a corporate head office where we were talking about the definition of cancer. It was definition of cancer. It's a private organization making very significant decisions within a small group of people, which I was part of. Fairly enlightened people, I thought, but the truth of it was that decisions made by that corporation made an impact on the kind of development and direction and protection and risk analysis just strictly on the basis of how the corporation decided to define that term. As is the same with disease. I work now with an organization where the definition of disease, from my perspective, is a pretty fluid one, but they have financial attachments and all kinds of decisions get made around their pretty conservative definition of disease. So these are very active issues, not just theoretical ones. They're continuing in the public eye without realizing it. And it would be interesting to me to know you, as people who are actively involved in this conversation now, are there efforts being made to, as you say, get the conversation started in the right place and the right hands? Lots of people have different ideas about how important whether it really applies or not. And I'm curious if there's any opportunities beyond attacking that have raised these issues in a more political.
So there has been a lot of discussion in the scientific community. So most of the meetings have happened in the US. It's something that is really on the radar of the scientific community. So there are experts that meet. They have met with concerned public citizens and government officials. Now, it's great. In North America, we may be very responsible with this technology. I think one of the problems is that this technology is so easy. It has democratized genome editing, but the drawback, of course, is that people outside of these jurisdictions don't have to play by those rules. So in the US, they've decided there can be no germline editing, so you can't change the sperm or the egg cells. So any genetic changes that could take place cannot be passed down to your offspring. So you're not allowed to make germline edits. But that doesn't mean that someone somewhere else isn't going to do it, or a biohacker in his or her garage. And the technology is incredibly easy to do. And I think maybe human genome edits, it's not really within the realm of a biohacker, because in order for you to be able to edit all of the cells in your body, you've got to be able to deliver to all of the cells in your body. So, there still is a very big hurdle to overcome. but certainly for engineering, super viruses. So the 1917 flu has been recreated using CRISPR. And I mean, I think beyond human genome engineering, that we really need to be thinking about the other viruses and bacteria and plants that we're engineering and where that could go. And I think those really provide the most dangerous use, I think, of, at least at this point in time, of CRISPR-Cas gene editing.
The decision not to involve genetics.
It's a regulatory, in the U.S. And many European countries also have adopted that. Canada, we cannot edit germline, but there are countries where they have not made that a regulatory guideline. So there are countries where it is legal to do.
I have sociological concerns about this new technique. It's advancing so fast. May I ask everyone, are we in the middle of a new revolution? 2009, we had the digital revolution. Now, with this velocity, this advance, we fourth revolution or
I think it's unanimous. Yes? Yeah or no? Maybe it's not unanimous.
Where's the ribbon?
There are things that we don't know yet. Oh, this is interesting.
Party's over.
There are things we don't. There are things we don't know yet, and actually, there's a paper that was published in the 5th of January this year, so 5th of January 2018, that describes actually an immune response by humans to Cas9. So also humans may have already developed system that allows them to somehow deal with Cas9. This was a paper that was published on the 5th of January. So there's a lot that we don't know about how, how organisms, because we've evolved together. Everything is connected in a way. We're talking about humans, but I think it's really important also to think about, we're not just doing this to humans. We're very scared about what we can do to humans, but I'm also really scared about what we can do to non-humans, to complex animals, and to really simple.
I could say, like, when you talk about revolution, I look at the history of all living things in humankind, even before humankind. And so if you look at a clock, as an analog clock, for example, as representing the history of time from the beginning of this planet, it's only around 8 o'clock that speech is and language becomes a global phenomenon, and then writing comes in around 9:30, and then printing, the printing press, and everything that comes with the printing press hits us around 10:45, and electricity hit us maybe at 11:30. And so we've been in this period of constant revolution, is my take on it. And it just depends on the lens that you want to, like, my lens is specifically about communication and those communication revolutions, but there are revolutions happening in small doses and all over the world every day, I would say. And then there's the longer look. So both and, again, is what I'm more comfortable saying to your question.
Humans are a revolution in the whole timeline
Yeah,
future
sure.
of the planet, right?
We've
been here like a second,
Close person, and then we actually close because there's the flow of meaning here, and for me it's a liberation. Like I want them to go home too.
my concern is twofold. One, you mentioned the word manipulation. You're an educator. I'm an educator. I see the purpose and the joy of letting people know. But there are situations where that mosquito unintentionally will have negative consequences. And then sometimes there are humans who have that mosquito in their head. Okay? And we're very good at disposing of everything. Okay? So this cure, which should be celebrated, sometimes some pharmaceutical might say, wait a second, but what happens to the cure that I have? My business will go out the window. I don't want that done. We can make a car that doesn't need gasoline. Scrap it. Because we want to sell cars, So you have all these industrial. We may be in North America say, our law says we don't do it this way. The British were great with, this is how we're going to fight wars. You put your line, I'll put my line. You fire your musket, I'll stand up. You sit down, I'll fire his, and so on. Until some guy decided, wait a second. Why not? We can kill them without the knowing. So some people don't go by the rules. How do we get this education? Everybody agrees. We're going to celebrate the cure. We're going to be knowledgeable that we are taking an awful chance. So when my wife and I decided to create a family, that first child was, we want to go through all that mechanism to make sure that he or she is perfect. If it looks like me, no, it's not perfect. But the idea was, and the question I asked at the time, because of the age, does it really matter? If it's not perfect, would you want to get rid of it? And she said, no. I said, well, let it go. Whatever comes. Okay. You just have to live with it. God knows the kid will have to live with us. So, I mean, how do we educate each other? How do we celebrate what you're doing? Keep in mind that the education piece is important, and yet still have behind our backs and behind our minds the fact that there's somebody out there that doesn't sing from the same song. And what do we do with them? Where's that genome or whatever it is to fix him or her?
Well, traditionally, we just kill those people.
Yeah.
Right? But seriously, like I used to teach in this small liberal arts college in upstate New York, and Blodgett Hall had the word eugenics carved into it for permanently. And then, they tried to put some arbor over it. But I knew what building I was walking into, the eugenics building. When the building was built in the 1880s, eugenics meant to keep a good household. And in Canada, we would call that like a chatelaine. But it's running the household. So what's good for us today might be bad for us tomorrow. And in fact, we don't have as much power as people who have more privilege than us. And we have more power than people who have less privilege than us. And so I want to ask the question, as an educator, are you aware of your privilege and are you using it wisely? I think that's the point of these kinds of discussions and this kind of work that you're doing. And I think it's what to celebrate. And I guess I just want to thank you all and thank you all for your attention.